Back. Okay. Good morning, everyone, and thank you so much for your time to come over here to attend this seminar or attending online. So we have multiple lab members who will present their research today. So we can go ahead and start. We'll try to make it quick and finish before we have pizza. Um, so I will talk about the research we are currently doing in my lab. These are all current projects. Um, but if you need more information, what we did before or what we are planning to do next, you can send me an email, and the email address is over here um, if you need more information. So because of time limitation, we will not present too many results in this presentation, but mainly the main idea is to show you what we are currently doing. And there are many opportunities for collaboration so that you know that what's going on between us and the industry and the USDA in the agronomic perspective for sugarcane. So just a brief introduction about sugarcane. Currently, we have roughly 414,000 acres. It went up in last couple of years. Sugarcane industry has a big economic impact of more than $3 billion. And most of this sugarcane area is in South Florida, around Lake Okeechobee, and mainly in Palm Beach, Henry, and Glades and Martin counties. 70% acreage on organic soils or muck soil, and 30% on mineral soil. The area on mineral soil or sandy soil is expanding. When I started this position, it used to be 80-20 split, and now it's 70-30. So it's going up, especially on fallow citrus land. So these are the only few production challenges. We may have a long list of production challenges, and there are always challenges in every crop. But this is related to what we are currently doing, what challenges we are trying to address in sugarcane production system. So one is like our new varieties whenever we develop, and normally then it's either infested by a new disease or pathogen or insect pest, or something happens so that we have to rotate it with another variety. So typically we have 7 to 8 years of life cycle, which is short. So we need to continuously produce new varieties so that we can replace the old one. Short crop cycle, we have 3 to 4 years crop cycle here, one plant cane, 2 or 3 ratoons. In some countries, it's up to 8 ratoons. In some countries, it's even more. So our main challenge is how we can further improve the ratooning ability and the crop cycle. Labor shortage, high planting cost. Planting cost is the major expense in sugarcane production system. So what we are in recently, especially after COVID, labor shortage is a major problem. So we are working on mechanical planting for that challenge. Harvest losses and damage to ratoon crops. There is high losses that when we do the mechanical harvesting, we have some losses at the time of harvest. And for that, we are trying to optimize row spacing according to our new varieties, which are high in biomass production. So talking a little bit about the sugarcane breeding program, this is a collaboration between 3 parties that started back in 1965. This, this collaboration being very successful so far. USDA takes the lead on that with the crossing is done in USDA at Canal Point. Seedlings, evaluation of initial stages, stage 1 and stage 2 on muck soil. Then it comes to us. University of Florida takes care of evaluation at stage 3. And then we also take care of some part of seedling evaluation on sand. Florida Sugarcane League takes care of evaluation at different stages. They provide technical support, they provide financial support. So they are the big collaborator on this whole program. So this is the scheme of the breeding programs. We have, previously we had only one breeding program, which was mainly for muck soil, but the varieties were mainly evaluated on muck soil for most of the evaluation period, but only at the last stage it used to be evaluated on sand soil too. But back in 2014, we found that we need to change that. So we have now two different programs, one on muck, one on sand. So these are the green boxes show like which are the initial stages, which are done at Canal Point. Then yellow is where we have on-farm trials on muck soil. All the orangish, brownish ones, they are on sand. So we have two parallel programs. All the crossing is done at Canal Point, and then at seedling selection, we move it to different places. So when you see in these columns, it starts at 80,000 clones, for example, in seedlings and muck soil, 1,000 on sand soil. But at every stage, we have a selection pressure, and then the number of clones go down, but we expand the number of replications or the plot size of that. So this is the agronomy lab employees who work under my supervision. I know most of you don't know everybody because not everybody sit in the lab over here. So we have multiple people work in collaborations with USDA, and they work with the scientists over there at Canal Point, and also with the Florida Sugarcane League. So University of Florida is involved in different uh, stages of the breeding program. And we have biological scientists, lab technicians. For example, in crossing, we have Maria and Rampasad Dayal. He's here today. Seedling stage on sand, Jairo is taking care of that with the help of Daniela. Jairo, Daniela, Ricardo, these are the ones you normally see over here, but many others who are here today, for example, Yasmin, Emily is here, all of them, they have big contribution to the program. So I would like to thank all of them because this is not possible without their help. They help in all the data collection. They do all the work in the field. And then all the graduate students, I have currently 4 PhD students, uh, one master student, Nick, who is working with Florida Crystals, and then Dayal, who is working with USDA. He's also doing masters along with that. So just wanted to thank everybody for their help on the breeding program. And then we have just briefly about what we do in stage 3 selection. What is it? What is it? So in stage 3 selection, we normally have 100 to 135 clones, which we plant at 3 different locations with 2 replications at each location on muck soil, and then 2 locations on sand soil, which is also 2 replications on each soil type. So then in plant cane, we collect some data on disease and do some Brix data. But then in first ratoon, we do all the yield data and harvest everything. Then we also collect disease data. So main selection is in the ratoon cane. Previously, we had it mainly in plant cane, but because of the importance of ratooning of sugar cane, we switched it to ratoon selection in different stages, including stage 2, stage 3. So out of 100, 135, based on first ratoon data, we select 30 to 40 clones. Then we collect data on those 30, 40 clones again. We combine data from first and second ratoon on yield and also on disease. And then we finally select 10 to 13 clones, which we advance to next stage, which is the final selection stage, is stage 4. At stage 4, then we have 10 locations. At each location, we have 4 replications. So selection procedure, overall, we want a high yielding variety. So our main selection is based on the economic index. We recently trying to update that. What economic index is based on the sugar yield and the current sugar prices. Based on that, you can calculate how much money you are making from 1 acre. And then we also include the harvesting and hauling and milling cost in that, and also the planting cost. So that helps in calculating like what profit you can get out of it. Because if you have high biomass variety, you may need more cost for harvesting that variety or hauling that variety. So other parameters are tons of cane per hectare, tons of sugar per hectare, sucrose concentration, then of course disease resistance. The major diseases currently we look at orange rust, brown rust, smut, leaf scald, mosaic. Then we have ratoon stunting disease, sugarcane mosaic virus, sugarcane yellow leaf virus, and we have Dr. Sushma Sood here. She takes care of mostly of the disease part. We look at all the plots in the field and do the natural infestation ratings, but then we also have artificial inoculation, which is done by Dr. Sood in the lab at Canal Point. So based on that, we release varieties every year, almost one or two, and sometimes up to eight. Over here, we have varieties that were released between 2005 and 2022. And you can see that there are the green ones are for sand, and then red ones for muck, and then muck and sand. Some varieties are good on both soil types. For example, even our current variety, which is 1201, which is our top variety, is grown on both soil types. Even same thing was for the 1252, which was our top variety previously, and now it's number two. So since this collaboration started, there were 125 varieties released. We also released seven or eight last year, which is not included in this. So there is a high productivity of this breeding program and the varieties we released. Um, how these varieties are used, especially in Florida, almost on 95% acreage in Florida. So this is based on the census we had last year in 2023. So we have 1201, CPCL 05 is number one variety. CP96 1252 is number two variety. So almost 50% or more acreage under two top varieties, and then the rest of the area is under different varieties. So we have a mix of new and old varieties, which are cultivated so that we can have the seed cane ready if we need to plant. Also, the impact of this CP varieties or the breeding program is not only within Florida. In Louisiana, they grow CP varieties. In Texas, they used to grow. It's just last year, this year, they just closed, shut down because of drought conditions. Internationally, these numbers are not up to date, but the idea is to show like the impact of these varieties in other countries. We have a germplasm exchange program, we have variety exchange program, so they grow our varieties too. If we look at the impact, so according to the data from the industry, Florida sugarcane industry, there is average increase of 73 pounds per sugar per acre per year in last 11 years in Florida. If we convert that into dollars based on currently we have 40 to 45 cents per pound for raw sugar. So that makes almost 116, $117 million additional income per year. And there are a couple of studies done. Uh, New varieties contribute about 60 to 65% of this increase in yield, but also the agronomic practices, environmental conditions, all these are also, they will also contribute. But varieties are the biggest factor. So we have, in addition to the breeding program, we have several multidisciplinary research projects going on. So one is mainly, as I said, we need better tillering varieties. So we are working on sugarcane tillering and ratooning. Hima is working on that project. She will explain about that in a minute. Then seed cane quality improvement for mechanical planting. We are working on mechanical planting in collaboration with the Florida Sugarcane League. We currently have trials with them. And if Simran is working on that, she's also a PhD student. Hima is a PhD candidate now. Um, He already passed qualifying exams. Then evaluation of row spacing in sugarcane. Kristen is working on that. She's also PhD candidate and working. This is her PhD project. New methods to collect sugar quality data. We have Emily today. She will present that data that we are working with the sugarcane industry to upgrade our data collection methods. Then evaluation of energy cane for bioenergy and ecosystem services. That's our bioenergy DOE funded project, which Sahel is working on. So he will present on that. So I would like to invite Hema to present her part.
Hello. Thank you so much, Dr. Sandhu, for the introduction. And good morning, everyone. And thank you so much for joining us today. Um, Hello. Yes. I don't know what's wrong with this one, I think. Oh, yes. So today I will be presenting my PhD dissertation titled Optimizing Sugarcane Tillering and Radooning Potential in Florida. Before we go into the objectives of my research, I would like to uh, give some introduction about what is tillering and what is radooning. Tillering, tillering is the ability of the plant to produce multiple stems from the base of the plant. Uh, these tillers are crucial because they contribute to the total biomass and thereby it has higher yields per hectare. Radooning, on the other hand, is the ability of the plant uh, to regrow after the harvest. This radooning capacity allows the sugarcane to harvest multiple times from a single planting. This will this will contribute or this will majorly impact reduce the production cost for the growers. Uh, Optimizing this tillering and radooning is crucial for the growers because it directly impacts their profits. Therefore, there is a need to understand the tillering and radooning, different factors that contribute to the tillering and radooning potential in Florida. So I have designed with objectives that okay, okay. I have designed the objectives to understand the genomic factors, physiological factors, and the environmental factors that influence the tillering and radooning potential. So with that introduction, let's move on to the objectives of my research. So the first objective, which focuses on the genomic factors that influence the tillering and radooning potential. So for this objective, we have selected two varieties with a contrasting characteristics. The the first parent, that is CP961252. This has a higher tillering ability, and the second parent, CP144588, it has a low tillering ability. These parents are crossed and the progeny is obtained, and this is evaluated. The progeny, along with the parents, is planted here at Everglades REC in an augmented randomized complete block design. And the morphophysiological, from this experiment, morphophysiological data, including the tiller count, plant height, SPAD, and the yield parameters, along with the genomic data with the help of molecular markers, will be collected from this experiment. The results from this experiment will help us identify it will help the sugarcane breeding program in marker-assisted selection to develop the varieties that have higher yields and better radoon crop establishment. So that's the first objective. The second objective, deals with the physiological factors that influence the tillering and radooning, and the aim is to quantify the different plant growth regulators in the tiller primordia. For this study, the same experiment site will be used, and from this study, the representative plants will be collected based on the high, moderate, and low tillering phenotypes along with the parents. So from these representative plants, the tiller buds will be removed, and these tiller buds will be used for quantifying the different plant growth regulators such as auxins and cytokinins. And the quantification is done with an advanced analytical techniques such as ultra high performance liquid chromatography techniques. So the idea is from this experiment, the results from this experiment will help us understand the different hormonal profiles in the tiller buds. Therefore, it helps us to understand the molecular physiology that is controlling the tiller formation and the development. And that's the second objective. And the third objective is dealing with the environmental factors that influence the sugarcane tillering and ratooning. The idea is to identify the best or the ideal planting and the harvesting times in sugarcane genotypes. For this objective, the experiment will be conducted here at Everglades REC, and three varieties are selected for this study. The three varieties are 1252, 926, and 1201. These three varieties are planted in three planting dates with four replications in each planting date. And the morphophysiological data will be collected, such as tiller count, plant height, span, and the yield parameters, as well as the soil moisture and the soil temperature data will be collected from this experiment for both plant cane and the ratoon cane. So the idea is from the results from this objective will help us to understand the different microclimatic conditions controlling tillering and ratooning, as well as to identify the ideal planting and the harvesting times, and also the sugarcane performance, the variety performance in each planting date. Here, I have designed, we have come up with this idea because we wanted to know the systems approach um, for improving the tillering and ratooning for the sugarcane um, industry. And yes, we are hoping to improve the tillering and ratooning potential. And if you have any questions about any other objective, feel free to contact me after the presentation. And I would like to thank Dr. Alia Momtaz from USDA, as well as the committee members and Dr. Sandhu for providing this opportunity to conduct this research. Thank you so much.
Next, we will talk about sugarcane planting system. Those of if you don't know how what's the planting time, what's the harvesting time for sugarcane, we have two different planting systems in Florida: successive planting, in which we harvest the last crop, last ratoon, and then we plant with the sugarcane again. And there is fallow planting or crop rotation, in which we um, harvest the last crop and then rotate it with some other crop, for example, rice or sweet corn, and or keep it fallow and then plant sugarcane again. So typically, our planting time is it starts from August till December. We have early sugarcane growth during January through April, and then grand growth period, which is currently going on May through August, and then harvesting starts again in October for every year. So we have different challenges because we have a short window of planting and harvesting. So harvesting is all mechanical, but planting is mostly manual. So during that planting window, we need a lot of labor to plant that manually. So currently we are working on sugarcane mechanical planting system, but there are different challenges which Simran will discuss, and her project is mainly based on that.
Uh, Hello, can you hear me? Okay. Thank you, Dr. Sandhu, for the introduction. And uh, I joined this agronomy lab last year in January, and so now it's been more than a year that I'm working in this lab. And I have right now I'm involved in almost all of the objectives that I have in my current project. And today I will be giving a brief overview about those. So starting with the title of my project, which is Systems Approach to Improve the Seed Cane Quality and Stand Establishment in Sugarcane. Basically, seed cane, which is also known as billets, these are the vegetative cuttings which are used for the planting of sugarcane. We obtain these billets or seed cane during the harvesting of the sugarcane using that, using a machine called chopper harvester, which harvests the seed cane and at the same time chops them into small billets. These are then transferred from this conveyor belts to a trailer, which is also moving alongside that harvester. And then once we have the billets with us, which are ready for the planting, uh, we use another machine called billet planter, which, uh, which conveys these billets from the wagon to the using a conveyor belt and then drops those billets into the furrows and at the same time applies these fungicides as well. Although mechanical planting have a lot of advantage, the growers can plant large areas in short period of time, but still these mechanical planting have a challenges such as the seed cane, it experiences damages, the buds are damaged, and those cuts and wounds during the transfer from one wagon to another wagon, from conveyor belts, the billets, they collide against each other and results in the development of low quality billets. Once these low quality billets are planted in the field, it results in the development of gaps or skips. So in order to avoid that, growers, they plant these billets at higher densities to compensate the loss. Also in general, the billets, they are, they have, they are more susceptible to diseases as they have greater exposed surface area as compared to the whole stalks. So in order to address those challenges, my research objectives were put forward, where we uh, address various aspects of seed cane, like seed cane preparation, where we plan to use various agronomic management practices to improve and standardize the seed cane quality. And second, in my second objective, we focus on the germination of the seed cane. Once the seed cane is prepared and we are ready to plant it in the field, how we can improve the germination of the seed cane. And lastly, last objective focuses on the protection of that seed cane from the various soil-borne pathogens that affect the germination of the seed cane. So under my first objective, we will be using different plant growth regulators, particularly growth retardants, so as to reduce the stalk height. The idea is that if we can reduce the stalk height, the idea behind the growth retardants is that they reduce the stalk height by reducing the internode length. Now the buds, they are grown on the they develop on the nodes, each node. So if we can reduce the internode length and we can have more number of buds in that standard billet size as compared to the non-treated ones. Here is a photograph of my experiment. This is one variety and this is second variety. In this variety, the effect of that growth retardant is more prominent. As you can see, we have some plots with shorter plant height, some plots with longer plant height. So that's the idea. And my second part under this objective is to apply the nitrogen before the harvesting of the seed cane. The idea is that just like any other seed, the seed cane is also dependent on its own food reserves for the establishment of root and shoot at the initial phase of its germination. So if we have well-nourished seed cane, it will have more probability to develop the roots and shoots as compared to the non-treated ones. And then my third part would be to see the effect of these two treatments in the subsequent crop cycle. Coming on to my second objective, where we plan to use various growth regulators and biological growth promoters for the germination of to improve the germination of the seed cane. So here we plan to use different growth regulators, apply them exogenously because growth regulators are required for the growth of any plant. And then in addition, we will use some biostimulants and growth promoters. Biostimulant like a kelp formulation, which is basically a seaweed extract, and it has been shown to improve the growth of sugarcane in many research studies. So we want to see if it's really helpful in uh, promoting the growth. And secondly, we want to use a growth promoter, which is basically a harpin alpha beta protein, and it initiates a systemic acquired resistance in plants, which is just like an immunity we get when we are given a flu shot. It is a weak protein which is found mostly in the pathogen, and once we expose our seed cane to this weak protein, it can induce our seed cane to develop some antibodies. So my coming on to my sorry coming on to my last objective, which is to optimize the seedkin protection against the soil-borne pathogens. This study is conducted under the guidance of Dr. Katia in pathology lab. So in this, we are finding different potential soil-borne pathogens which might affect the seedkin germination. Basically, we look for the gaps in the fields or some diseased plants, and then we dig out those canes, and then we split them open to see if the cane is diseased, and then we try to get the samples from it and grow the pathogen in the lab and then identify them. And then later we identify them at genetic level to differentiate between the strains. Once we have different strains, then we can use those strains to, again, infect the sugarcane in the greenhouse so as to confirm that those isolates are really the pathogens for the sugarcane. So once we have those pathogenic strains with us, we can later use it to screen the sugarcane germplasm to find the resistant cultivars. And with that, I would like to thank Florida Sugarcane League for funding this project, and Florida Crystals and US Sugar for providing us the land for the experiment, and the crew members, and also the committee member from University of Florida and Australia, Dr. Troy. And then I would like to thank my lab members from Agronomy Lab and as well as Pathology Lab. Thank you.
Thanks, Simran. So next we will move to the row spacing impacts. So this started, we did some research previously in small plot studies over here at EREC. We saw some interesting results. Industry also tried to experiment on different row spacings in commercial planting. And then this idea came in last year that we should look further into it. And this year we have one research trial with Growers Co-op. And that's what Kristen will talk about, and we will expand on this as we move forward with this research.
There's the, okay. Hi, guys. Hey, I know it's good to be back in the sugarcane industry. I took a break, but I'm glad I'm back and in student capacity and USDA capacity. So as Hardev explained, I'm going to be looking at row spacing, and the title for my project is Determination of Row Spacing Impacts on Sugarcane Yield and Ecological Parameters within the Everglades agricultural area. So I'm actually really excited about this project because I'm an ecologist by trade. All right, so the concept behind this project is that row spacing is variable throughout sugarcane production regions. Regarding planting and mechanized harvesting, I can kind of, piggyback really off of what, previous student was doing. Anyway, and then traditional spacing in the EA is 5 feet, but it is shifting towards 6 feet. I know when I was with the co-op, we were looking at 5-foot spacing and 6-foot spacing at a commercial level when I was there last year. And then little research has been done on variable row spacing within the EA and its impacts on high yielding varieties. Okay, so I have 2 objectives, and this is all just, it's developing work in progress, so these could change. And first one is to determine how increasing row spacing from the standard 5-foot centers could help reduce soil compaction, harvest losses, and production costs. And then determine ecological impacts increasing row spacing has within sugarcane production and how that affects parameters such as plant nutrition uptake, disease pressure, weed pressure, canopy closure, and yield. So this is my study site at Sugarcane Growers Cooperative on Glades Sugar Farm. Elliot Rams, who is the farm manager, put this in, and I'm going to be putting plots in. So it's about 33.1 acres, a second site to be determined. So as you can see, there was a 10-foot alleyway that splits the 2 varieties. So we're using CP06 2042 and then CP-CL05 1201. There are 3 treatments, a 5 foot, 5.5, and 6 foot center, 2 reps each, and then 12-foot alleyways. And the treatments are 15 rows across each. And then I'll be putting subplots into that, 4 rows across, 5 plots per treatment for 60 plots. And that could change too. It's kind of a work in progress and everything. So I'll be looking for feedback, suggestions, anything like that. And then sampling from the middle rows, which is industry standard. All right, so parameters to be measured, yield and growth, looking at tillering, stock weights and commercial yield, and doing some CPS work, that type of stuff. With plant nutrient uptake, leaf tissue analysis, I'll be going out collecting leaves and seeing how the different row spacing affects that. Canopy closure timing, like light penetration, using sun scan, and all that stuff. Weed pressure, looking at the species present, percent coverage, how often are we going out and treating those weeds with herbicide application? Disease pressure, going out looking at disease ratings, looking for rust, looking for smut, that type of thing. And then repeating it for the retuning, and then also looking at soil compaction, et cetera. So that's pretty much that in a nutshell. Again, it's kind of organically working through things, but I just want to thank my committee, Elliott Rounce, for putting in the project at his site, Florida Sugarcane League, and USDA ARS. So.
Thanks, Kristen. And special thanks to Elliott to make it happen this year. So we'll continue this in next couple of years. So the next one is we continuously strive for improving the methods of our data collection. And this one, which we are looking currently, will be game-changing if it will work. We have in sugarcane, the people who work in sugarcane, you know how hard it is to collect all the data, especially the yield data and sucrose quality data, because you need to harvest all the bundles, bring them back to the lab, and do all that work, which need, even just running the CPS, you need 6, 7 people at a time. So Emily is currently working with Yasmin and others, and especially the sugarcane industry is helping in this. Mainly leading this project, which we are conducting over here at EREC and also at USDA Canal Point. So Emily will talk about this briefly, what she's working on.
All right, hi everyone. As Dr. Sandhu mentioned, we're looking at an easier methods to collect the data for sucrose analysis, and so the micro NIR has been what I've been working on for the last year or so. It's a handheld field portable near infrared spectrophotometer that we're using kind of with the CPS just to compare what methods are, if it's a suitable method to maybe replace it in the future. Because like he mentioned, the CPS takes a lot of manpower and it's also destructive. So as we go through the slides, you'll see, I'll give you a brief overview of both the CPS and the micro NIR so that you can see the difference in the ease of use with the micro NIR versus the CPS. But both of them we're using to measure Brix, poles, moisture, and fiber. So the first step is harvesting the sugarcane, which usually happens the day before. And we decided on varieties to use, like specific ones that we know the sugar of, and we're going through the season from low sugar to high sugar. So we can kind of use those as a, I don't know, reference. So yeah, we'll determine the varieties, and then it's we harvest 5 stock bundles per each variety, and based on, because we're doing wet chemistry to get the data to use for calibration, so based on how long it takes us to do the wet chemistry, we usually do around 20 to 21 varieties a day, because the wet chemistry is a long process. And I forgot to mention, but in the other slide, this is a much quicker than another traditional method, the wet chemistry, but we're using that to collect the data so that we know that it's right for making the calibration curve. So step 2 is when we're using the micro NIR, and you can see here we first bring the stalks in the lab, and we have to reference the instrument, and so we used between like 5 and 7 micro NIRs that we accumulated throughout the season that we're trying to get enough data, spectral data for to create a good calibration curve. And how do we do this? Okay, so here we're using the tough books, which are rugged so that we can bring them in the field. And however, there is a app underway that's in a prototype right now that will hopefully make it more convenient, because when using these in the field, you can get about like a truck length away from the tough books with the instrument before the Bluetooth disconnects. So we have to reference the micro NIR, and there's not a picture, but we had developed a nose piece that fits on it, so it can the piece is flat, and so putting that against the round sugar cane, it doesn't really like stay in place. So they developed a nose piece so it can like sit against the cane and not move around for a better measurement. And then since we're using multiple instruments and we want to mark them in the same spot, measure in the same spot, we mark it, and our like reference point for marking it is between the 4th internode from the top and the middle, just because it's the most like representative spot to measure. Then we scan the marked areas on all of them, which takes 1 to 3 seconds depending on how it's set up, which can be adjusted. And then we export that spectral data. Next, we, again, like I mentioned, we're doing CPS and micro NIR at the same time, so we can get a bunch of spectral data so that we can build a good calibration curve for both instruments. And so yeah, this is the one that's kind of a lot more labor intensive. So first we have to, you'll see in the pictures, we have to shred the cane, and then and then over here we have to load it in, and then it comes out here, but it takes quite a bit longer because it's reading the shredded cane in multiple different locations, as opposed to the micro NIR is just reading in one spot. So it takes, I don't know, 20 seconds to run through. And then when it comes out, this is where we take a subsample of the shredded cane to bring to the lab for the wet chemistry analysis. Over here, you can see that the data comes out on a computer. So again, you need someone running the computer, whereas the micro NIR, you can have real-time data availability right there on the laptop or the app on the phone once that gets finished. Yeah, so this takes about, I don't know, five people usually, and then plus harvesting in the field. Then we take the subsample to the lab. I'll just go to the pictures and explain it. So first we weigh out 500 grams. That's what we determine for the calibration equations that we're using. And then over here we have the hydraulic press, which presses all the juice out, which we collect, and then left in the bottom is like a plug of the shredded cane, which we then weigh and put in the oven overnight. So that's used for fiber calculations. You can see there we put them in a sieve so that the airflow can go in the convectional oven. And then with the juice that we take from the press, we measure the bricks and the pole. And so here you can see that's our setup for measuring the poles. We clarify the juice with Octopole and filter it through the funnels, which that is also pretty time consuming. And then we measure that over here on the polarimeter, and then right here is the bricks measurement. So we take a little bit of the juice from what came out and spin it down in the centrifuge for about a minute, just to collect all the debris at the bottom, so it doesn't affect our readings, and measure that on the refractometer to get the bricks. So yeah, right now I don't have any like data to present because that's still in the works. We probably had about like around 700 points of data points that are being used to build the calibration curve for both the micro NIR and the CPS. And that will be presented probably at ASSCT. But yeah, just a quick overview. The micro NIR is quicker, and you just take it out into the field and measure the and scan the stalks versus having to harvest and shred and run through the CPS. And you see all the work that goes in with the wet chemistry, which we can only, again, do about 20 samples a day, which will be necessary for calibrations every year. But as a overall method for getting the parameters that I mentioned, it's not really feasible. So thank you.
Thank you very much. So our last part is mainly about the bioenergy project we are working on. We will quickly go through this in the last few minutes. So bioenergy research was started in parallel to our sugarcane breeding program. It started back in, I think, 2005, 2006, when we started looking at some of the varieties which are high fiber varieties, but low in sucrose varieties. So there was a big push from the industry, mainly from the BP, British Petroleum, and some other industry to look into the biofuel crops. At that time, we tested different biofuel crops, and energy cane came out one of the most promising in Florida and also in the southeastern United States. Then we developed 5 varieties in collaboration with USDA ARS Canal Point, and we released these varieties for commercial production. Currently, we have these varieties over here. And then our current project is mainly focused on the evaluation of energy cane for bioenergy and sustainable agriculture systems. This was funded by DOE back in 2021, and this is a 5-year project until 2026. We have collaboration with Argonne National Lab. We have scientists from here, from UF and Argonne National Lab, and then also the collaborators from aviation fuel industry and others who are working with us on this. So the project overview is mainly like we have citrus production, which is going down with time because of citrus greening disease. So what we have is this in 2022, it's almost 200,000 acres of citrus fallow land currently. So we are looking at if energy cane can be an alternate crop on marginal soils where we can produce any other crop. And also in sugarcane area where sugarcane can't be produced anymore on shallow soils, and energy cane is more efficient in nutrient uptake and use of water resources. So maybe energy cane can be incorporated in those areas. So mainly we are working on field scale production system. And also this data then goes to Argonne National Lab, and they work on mainly machine learning and remote sensing, and also to look at the techno-economic analysis, life cycle analysis, and all that. With that, I would like to invite Sahil to present his project.
Thank you, Dr. Sandhu, for the introduction. So the topic of our research is evaluation of energy cane for bioenergy and ecosystem services in Florida. So as Dr. Sandhu already gave the overview, I'll start with the objective of this research. So our first objective is to evaluate energy cane for seasonal biomass accumulation and nutrient uptake. As we are trying to integrate energy cane on marginal and fallow citrus area, as Dr. Sandhu mentioned. So our hypothesis for this is that integrating energy cane on marginal and fallow land will produce enough biomass and will uptake more nutrients than the other crops. So second objective is to compare the effect of energy cane on soil health and quality. For this objective, our hypothesis is that energy cane integration on these areas will reduce the soil water nitrate concentration and will improve the soil chemical and microbial properties. Third objective is to determine the effect of energy cane on water quality and quantity. For this objective, our hypothesis is that the energy cane will improve the nutrient loading into the water bodies. Our last objective is to determine the effect of energy cane on soil biodiversity and wildlife habitat. For this, is our hypothesis is that the integration of energy cane will not have any harmful effect on these biodiversity. Other than this, we are also working on greenhouse gas emissions, and Dr. Darub's team is taking care of that along with Pamela. So for this project, we have two experiment sites. First is on the organic soil. Here we have three crops: sugarcane, energy cane, and sweet corn. So here we are comparing energy cane with sugarcane and sweet corn for ecosystem services like water quality and quantity, soil health, biodiversity, and greenhouse gas emissions. So on the experimental plot, these marginal areas were identified. As you can see, the shaded ones are the marginal areas, and the energy cane was integrated on those marginal areas, while the sugar cane and sweet corn remains on the fertile areas. After planting the cane and sweet corn, so we apply 2 nitrogen and 2 phosphorus treatment. Since energy cane uses the fertilizer recommendation of sugar cane, as energy cane has no fertilizer recommendations on this location, we added the fertilizers with the recommended fertilizers. Second experiment site is sand soils at Indian River Research and Education Center in Fort Pierce, which is a fallow citrus area. So here we are trying to compare energy cane with the fallow areas for ecosystem services. So on sand soil also, we use the fertilizer recommendation of sugarcane. But on this location, we reduce the fertilizer treatments by 50%. So because our hypothesis is to see the nutrient response on seasonal biomass accumulation and nutrient uptake on both the locations. Data was collected during different time of the years for different parameters. So currently we are in the first ratoon at both the locations, and we already finished with the data collection of plant cane. For instance, for biomass quantity, the cane was harvested monthly, and after taking fresh weight, the cane was chopped and dried for dry biomass accumulation. We got dry biomass yield of around 45 megagram per hectare or on organic soil, where treatment one was the recommended fertilizer only, and treatment two was the recommended fertilizer plus additional nitrogen, and treatment three was the recommended fertilizer plus additional phosphorus, and treatment four was the recommended fertilizer plus additional nitrogen and phosphorus. So here we found that the nutrient application has no significant effect on the dry biomass yield. On sand soil, we got dry biomass yield of around 22 megagram per hectare, and here the treatment one was the recommended rate, and in treatment two, the phosphorus was applied at the recommended rate while nitrogen was reduced to 50%. In treatment three, the nitrogen was applied at a recommended rate while phosphorus was reduced to 50%. And in treatment four, both the fertilizer was reduced to 50%. And here we found that the nutrient application has no significant on dry biomass yield. So we got the similar yields with the reduced fertilizers, which meets with our hypothesis. So these are the data from the plant cane. So we will confirm these statistics in the first and second ratoon. And I would like to thank Dr. Sandhu and our agronomy team for helping us in collecting these data. And due to time shortage, we were only able to present this much of data. Thank you.
Thanks, Sahil. So next, just quickly summarizing, I know it was a lot to cover in 1 hour. So in summary, I would say new varieties are very important in sugarcane, but we can't ignore the management practices. We have to keep updating the management practices with the new varieties. Multidisciplinary research is important to address the key challenges, and then collaboration is the key to success. So that's what I'm doing for last several years. and it helps you a lot to conduct the research successfully, to do the multidisciplinary research project successfully. So I'm always thankful to all the collaborators who work with me on these projects. And at the end, I would like to acknowledge the support of all the local and at national level, especially Florida Sugarcane League. For Florida Sugarcane League is sponsoring a lot of the employees who are working on the breeding program, and also funding some graduate students to work on different aspects of the production system. And also USDARS, this collaboration was going on since 1965, and I wish it continues forever. And it's always pleasure to work with the scientists over there. Individual companies, Florida Crystals, U.S. Sugar Corporation, Sugarcane Growers Cooperative of Florida, always willing to help in providing in-kind support too, so that we can conduct research trials on their farm, and they manage our trials. Department of Energy for providing funding on bioenergy, Argonne National Lab working with us on that project. And thank you all for attending this seminar. We can have some quick questions, maybe 5 minutes. Avery?
I haven't got that far yet. I'm currently actually writing my proposal. So Hardev was kind of like, hey, come give a presentation. I'm in the moment I'm writing the proposal. So for the microclimate stuff, I'm mainly after the bigger picture stuff. So like your soil compaction, everything. But for like microclimate, like relative humidity in canopy temperatures, just, simple hobo loggers would be easy. Just place them out there. Easy peasy lemon squeezy. So yeah, but that's a good point. So if you have any recommendations, send them my way.
Yeah, if we will include the disease part on this, I think we will need a pathologist on board and work with us mainly on the microclimatic conditions for rust. So we'll plan for that. Any other questions? Yes, Sushma.
Control.
Yes, the treatment one was the recommended fertilizers, that one we use on muck soil and sand soil.
Yeah, I was going quick, too quick. Any other questions? Okay. Yes, of course.
What's the variability from the micro NIR from two consecutive measurements from the same point?
You mean if we measure the same?
Yeah,
same-
twice.
Very close.
Very close.
Yeah.
Okay, thank you.
You will see some results if you're attending ASSCT meeting next month. Yasmin is going to present a poster on that. But it's still the first year of data collection. By next year, I think we will have a lot more results to present on that. Okay. Yes, Wayne. Yes. The outlook now is to look into different aspects, especially the aviation fuel. Previously, we were just looking for ethanol production, but now aviation industry is much interested in biofuel. And they already have some collaborations in Australia with the sugarcane industry over there. And also not just biomass from energy cane, but any other biomass they can use. And the idea behind this now is also to look at the ecosystem services. If we can improve the soil quality, or if we can grow that on marginal land where the commercial food crop production is not possible or feasible. So if we can add all that and make sense out of it, I know the ethanol production was not very economical, and it didn't work. So over time, I think the main idea was like how the science or scientific information changed, technology changed over time to produce ethanol from lignocellulosic material. But also the aviation fuel industry is the major, I think, push behind this now. Yes, one.
Can you hear us?
Yes.
Yes. Okay. We have a few questions online. I don't know if you want to go through, if you have time. Sorry for
Okay.
interrupting.
Over here. Are there process-based modeling research effort within the UF sugarcane group? I know there was some modeling studies. Previously, there was some research done. Jim Shine was working here, and he was collecting data for his PhD to do the modeling stuff. And on that, we are working with different countries, different sugarcane producing countries, and there is a consortium which is working on this. And another question is, how is germplasm collection for bioenergy crops? So yes, we have, we didn't actively work on the breeding program for energy cane since long, but we have some good germplasm available. And there was actually, this research was initially, it was funded by BP, British Petroleum. And then, as Wayne mentioned, that it didn't make sense economically, so that research didn't go through after that. But we have germplasm available for energy cane, and that can be further tested if needed. But at this point, I think the Department of Energy and other agencies are mainly looking into not only just the variety development, but also mainly the ecosystem services part of this. Okay, there's no other question.
Okay.
Oh, yeah.
All right. I have a question on the study that you conducted in the sandy soils for bioenergy production. Normally, I think growers, they put a lot of fertilizers in sandy soils because of leaching. But you said you applied the recommended rate for phosphorus, but you reduced nitrogen to 50%, but still mold, you didn't have like effect on the biomass production. Why would that be so?
So your question is that why we reduce the fertilizer rate?
If you have identified or determined why the biomass still remained to be not affected after reduction of nitrogen application to 50%.
Okay. The energy cane has a very good root rhizosphere. So it has very long roots which can penetrate deep into the soil. And these roots can uptake the more nutrients as because energy cane has more nutrient use efficiency. So that was to check the nutrient use efficiency if they can uptake more nutrients efficiently. So that was the reasons the yields will remain same. And also they will improve the water quality because we are adding less fertilizers to the crop.
Also, the recommended rate was for sugarcane because we don't have any recommended rate for energy cane. So that was not a recommended rate for energy cane. Thank you for the question. Okay, you can have one more question because we are getting late for the pizza, I think.
Thank you for the presentation. I would like to ask Sim, I guess, how much will cost implement mechanic planting compared with manual? How much will cost implement mechanic planting compared with manual?
Because initially the problem was mechanical planting that had extra cost that so much data yet. Thank you very much. Please don't forget to